Welcome to Chapter 2 of AP Psychology Methods. So, for the intro, first we have to look at hindsight bias. Hindsight bias is a tendency of people upon hearing about research findings to think that they knew it all along. For example, if you told someone that married people tend to live longer, the person would say, "Oh, that's obvious. Everyone knew that," even though if you didn't know that information before, it might not seem that way. So things sound obvious. And then we have to look at the difference between applied research and basic research. So, applied research is where psychologists conduct research to solve more practical problems. For example, seeing how personality might interfere with workplace relations. So, this is something more practical and has a direct application in our world. It is something that perhaps you and I would be more interested in. However, there's also basic research, which is research that answers questions that probably only psychologists really have an interest in. For example, they might be looking at the brain, vision, how perception. This is research that might not immediately have an application in our world, but it is necessary for psychologists in order to advance their studies. And then we look at the actual research process. So in research, first we have to come up with a theory. So a theory aims to explain some phenomenon and allows a researcher to later generate a hypothesis in order to collect data that might support their theory. To support. And then, so a hypothesis is a statement that expresses a relationship between two variables. And the variables in this case would be your independent and your dependent variable. Your independent variable is a variable or the factor that you don't change, while your dependent is the factor you do change. And by analyzing the relationship between these two, you can reach some sort of conclusion about your research. And then we have in research your operational definitions. Your operational definitions define how we measure the variables. So operational definitions are necessary because we want to ensure that every future researcher knows our steps. And this is necessary in order to make our research reliable and therefore repeatable. For example, if you were measuring stress in our research, we will want to note which type of machine we use so that if someone were to try to repeat our research, they would use the same machine in order for our results to be more comparable. So the two factors that we have to take into account when we do research are validity and reliability. As I stated previously, reliability is how repeatable your experiment is. If it was something that maybe only you could have done, or something that is just very hard to measure again, it is not a very reliable research, and generally, the results you get are not going to be considered very valid either. So, for validity, research is considered valid when it measures what the researcher set out to measure. And then we move on to the methods of research. So, the first thing we have to take into account when we are doing research is our population. The population is the group of people that we are researching or experimenting on. The idea of getting a population is to try and make it as representative as possible, which means that from looking at maybe about 100 or 1,000 people, you want to be able to gain the general picture of society. So for a population, you probably don't want a population of all girls or all boys or a population of only one ethnic race. You want all the factors of society mixed into those 1,000 people. And the way we ensure this is through sampling. First, there is random sampling, which is the preferred method. This is where we just randomly pick about like however many people we need out of our overall population. And then there's stratified sampling. Stratified sampling is similar to random sampling in that there's still a certain amount of randomness in it. However, stratified sampling looks at the percentages in our population and takes your population for your research based off of that. For example, if you were doing research in a school where there was about 1,000 kids and you had 30% who were African American, 20% were Latino, and 50% were Caucasian. If you were to try and get a sample of 100 students, stratified sampling means that you would get 30 African Americans, 20 Latino, and 50 Caucasian. And now we look at the various types of research. So first, there is case study. So a case study, as I will write up here, is a study of one specific person. For example, the famous study of Phineas Gage, where we look at someone who has some very unique or different behavioral or psychological trait, and we try and analyze that person in detail over a long period of time in order to get a deeper understanding of their unique psychology. So in Phineas Gage's example, he lost a large portion of his brain in an accident, and yet he was still alive, but his behavior had changed. So by looking at him, we can maybe see how our brain influences our behavior and how exactly he managed to survive. The good thing about case study is that we can gain a lot of insight into some more obscure aspects of our psychology. However, a case study is non-representative. As you're only looking at one person who is actually quite unique in the realm of psychology. They don't represent the rest of society. Not everyone had Phineas Gage's problem, and probably not everyone would have survived as he did. And then there is naturalistic observation. So naturalistic observation is where we look at a person or animal in their natural habitat. The benefits of this type of research is that they act completely naturally. In comparison to if they were in a lab and being asked to do all sorts of tests, you might not get their most normal behavior. However, the bad thing is that when we are out in nature, out in the world, there are many more factors that we have to take into account. In a lab, everything is closely monitored and controlled, but out there in the world, it is much harder to do so. So you might have some confounding variables, as we will talk about later. And then finally, there is the survey. The survey is, pretty sure everyone has done a survey at some point in their life, where you just get asked many questions, and then you submit your answers to some sort of place. The good thing about a survey is that you get pretty much a random population, which is obviously the preferred method, and you also get a really large population. It is not that hard to send out a survey to many people. And then by having this, you have a lot of data, which makes it easier to support any correlation you come up with. For anyone who has done statistics, obviously more data is always preferable to less. And then we look at confounding variables. So a confounding variable is any difference between the experimental and control conditions except for the independent and dependent variable. So in short, they are factors that we didn't take into account or just couldn't control. And these are generally the reasons why some of your research data might be a bit skewed or not in favor of your hypothesis. And then finally, there is assignment. Assignment is a process by which participants are either put into an experimental or control group. A control group is generally where we set a baseline. So in the idea of clinical trials, if you are familiar with that, there's the idea of a placebo where you give some people a fake drug and then you give people the real drug. So the fake drug would be the control group. The control group just sets the baseline. What is the normal human reaction? And then from that, we can see how different the experimental group's reactions are.
Okay, so moving on to bias, you have two types of bias, experimenter bias and participant bias. So for experimenter bias, the experimenter will try to treat the subjects differently depending on whether they're in a control group or observed group in order to prove their hypothesis. And then you have participant bias, which is when a participant know whether they're in the control group or observed group, they'll act differently based on what they're given. So in order to avoid these two type of bias, we have what's called the double blind procedure. Remember, make sure you know this because it's really important. In order to avoid any sort of bias by the experimenter or participant, you have this procedure in which you randomly assign people to control group or observed group, and neither does the experiment nor the participant know which group they're in, so which avoids any types of bias. And then you have the Hawthorne effect, which is also avoided under the double blind procedure. Hawthorne effect is basically people's tendency to conform and please the researchers. So if the researchers want their hypothesis to be a certain way, the participants will act in that way in order to please the researchers. Now moving on to the analysis of statistics. So you have a scatter plot, which is when you put all of your statistical data onto a set of axes and then you draw a line of best fit in order to measure the relationship between the two variables. And then you have -1, 0, and +1. -1 means you're perfectly negative, so as one increases, the other decreases. 0 means that there's no relationship between the variables, and +1 means that one increases, the other one increases too. So descriptive statistics is your way of describing a set of statistics. So there are two ways of describing them. First,
No, but it's continuing. No, like it breaks it up.
are you sure?
Yeah, I know. I got it like that.
Okay, so you have analysis. So once you get your statistics, you're going to want to analyze the statistics. So firstly, you have a scatter plot. So you plot all of your numbers on a set of axes. And then you draw a line of best fit that represents the relationship between your variables. So your statistics is going to be close to one of these three numbers, -1, 0, and +1. So -1 means a perfectly negative relationship. So as one increases, the other one decreases. 0 means that there's no relationship whatsoever. -1 means that there's a perfect positive relationship. So as one increases, the other one increases as well. So for descriptive statistics, this term means that it's ways that psychologists use to analyze data and describe it. So you have central tendency. Central tendency is your mean, median, and mode. Recognize that for the majority of times, these three variables are going to be different once you get your numbers. So for every set of data, you always have what's called the outlier. Outlier is a number that is larger or significantly smaller than all of your other numbers. So there are two types of graph that you need to know. If number is larger than all the other ones, the set of statistics is positively skewed. Positively skewed means that your mean is going to be more than your median. It will look something like this. So your mean is going to be higher, so it's going to be here, and then your median is going to be up here. And then if the number is smaller than all the other ones, you have a set of statistics that is negatively skewed. And it's going to look something like this. So if it's negatively skewed, your mean is less than your median. So your mean will be here, and your median will be here. And the other way to describe a set of statistics is called variability. So variability has three components. You have range, variance, and standard deviation. So range is the largest, subtract the smallest. The variance is the distance between every number to the median number. And your standard deviation is the square root of your variance. And you don't need to know how to calculate them, just know what they mean. This is a normal distribution curve. And we can see that for the majority of the experiments that are done by psychologists, 68% of the numbers fall under one standard deviation, 95% of statistics fall under two standard deviation, and then 99% of the statistics fall under three standard deviation. So inferential statistics. So we know that descriptive statistics is ways that we can describe a set of numbers. However, inferential statistics is how relevant is this experiment? Is it statistically significant or not? This means that whether or not the statistics are the way they are because of chance or because there's an actual relationship. So you use what's called the p-value. The p-value shows you that if it's within 5%, this is statistically significant. If it's larger than 5%, it's statistically insignificant, which means that it happened probably by chance. So once you decide that you want to do an experiment, you apply to the APA, and they have a set of guidelines that you need to follow if you're using humans or animals. For humans, you have 5 rules. Firstly, no coercion. You cannot force someone to do something. They need to voluntarily sign themselves up for your experiment. They need to have informed consent, so they need to give you consent, and on the basis that they know what's going to happen during this experiment. Confidentiality is really important. You cannot expose their names on the internet or any other sort of mediums. And fourth, you have lack of risk, so you cannot put humans in mental or physical distress. And at the end of the experiment, you need to have a debriefing process in which you give the participant your phone number, your email, so they can contact you if they have any questions after that. For animals, you have less restrictions, but you still need to follow them in order to get approved. So firstly, you need to have a clear purpose. You can't just buy animals for no clear scientific purpose. You need to have a proposal that outlines your experiment and what you're going to do with these animals. And they need to be cared in a humane way. So outside of the experiment, you need to give them food, water, and whatever keeps them alive and comfortable. They need to be acquired legally, so you cannot buy these animals from a black market. They need to be legally acquired. and least amount of suffering. So even though that there's going to be some kind of uncomfortable variable within the experiment, it needs to be overall the least and the most minimal amount of suffering that the animals are exposed to. So this is everything you need to know for chapter 2 of AP Psychology. Good luck in May.